Hello, we will continue to discuss a particular point of the temperature-dependent sex determination in turtles, which is what we can do for intersexes or ovotestis. I will explain the difference. First, I will recall some basics information about temperature-dependent sex determination in turtles. So the typical pattern for the species is male at low temperatures and females at high temperatures. An individual is defined as a male if it has a gonad of testis type. It is a female if it has a gonad of ovary type. During development, the gonad is first non-differentiate, then it acquires some characteristics of males or females, so testes or ovary. It is nonsense to say that males gonad differentiate before females gonad because this is the same organ, and if it is not a male gonad, testis, it is an ovary gonad. So you cannot say one differentiate before the other. Typical testis looks like this kind of gonad. The structure is typical, in this case, with wide tubes or cord present in the medulla, and the gonad is covered by a very thin epithelium. In the adult testis, the medulla consists of seminiferous tubules formed by Sertoli cells and germ cells. These tubules are surrounded by myoid cells, peritubular cells, and Leydig cells. A typical ovary is shown on this graph. The medulla is formed by mesenchymal tissue in which thin epithelial cords or lacuna limited by flat epithelium are observed. In the cortex, oogonia in meiotic prophase and growing oocytes are present. This is shown by the C letter in this histology photograph. In the mature ovary, the medulla consists of mesenchymal tissue with lacuna of varying size depending on the species, and on the surface of the ovary, the cortex contains follicles, oocytes surrounded by granulosa germ cells and thecal cells. So we have seen typical ovary and previously typical testis. We have seen a typical testis and a typical ovary. Here it looks like a typical testis, but look at the cortex at the right of this gonad. You see some oocytes that are present, and this gonad could be considered as a ovotestis because it has typical tubules in the medulla, but still oogonia in the cortex. It looks like a testis with a little part of cortex. This gonad is typical ovotestis. The individual is named intersexes, but the gonad is ovotestis. You see in the medulla well-differentiated testicular tubes. Wide testicular cords are present, but more than half of the surface is covered by an ovarian-like cortex. This gonad is a typical true ovotestis. To understand how it can happen, so we summarize what we have seen previously. The medullary part of the gonad, so the center, is organized as tube in testis, whereas the epithelium stays not organized in ovary. The cortex or periphery of the gonad expands in ovary, whereas it remains very thin or it degenerates in testis. Both these phenomena are mediated by the level of the steroid hormone estradiol 17 beta in the gonad. Take a look at the graph in the right. I have tried to summarize what's happened for differentiation of the gonad depending on the level of estradiol 17 beta in the medulla at left and cortex at right. When the level of estradiol 17 beta is very low, it permits the proliferation of tubes in the medulla, and the cortex stay very thin, it does not proliferate. When the level of estradiol 17 beta is very high, it blocks the differentiation of tubes in the medulla, but it permits the proliferation of the cortex. You have an intermediate level of estradiol 17 beta where the tubes are not blocked, differentiation of tubes are not blocked, but the cortex is, you have a signal to enhance the differentiation, the proliferation of the cortex. And then you have, in this case, ovotestis. In some species, nearly all the testes show some level of intersexuality in the gonad. For example, the leatherback turtle, Thermochelis. In others, the ovotestis are frequent. For example, in Emys orbicularis, the European freshwater turtle. And still in others, it seems to be rare or absent, for example, Trachemys. The proportion of testis gonad covered by a developed cortex varies a lot, both from anterior to posterior part of the gonad, but also between individuals. An experimenter could decide to classify an ovotestis as a testis because of tubes in medulla, or as ovary because of developed cortex, or as an ovotestis. This is a matter of choice depending on the sensitivity of the experimenter. Intema suggests that intersex gonad differentiate into ovaries in adults. However, in many cases, individuals with this ovotestis have degenerated Mullerian duct, so they cannot be functional females. Pio considers more probable that ovotestis developed as testis, and we will keep this solution, which are more logical. So in conclusion, the presence of ovotestis is common in some species of turtles with temperature-dependent sex determination. The classification of a gonad as an ovotestis is highly dependent on the personal judgment of the experimenter. When an ovotestis is noticed, I consider more probable that it will resolve as a functional testis, even if some developed cortex could persist at adult stage. Thanks.